పాతిట్లనే ఓకే మేడం.
Good morning everyone. Now going to today topic is the C4 cycle. Then the C3, C4, CAM. These are the different types of the pathways of cycles. So now in previous class we have to completed that the C3 cycle. Now I am going to the C4 cycle. Okay. Then the C4 cycle is also called to the Hatch-Slack cycle. Hatch and slack. Hatch-Slack cycle. Because of these are the both colleagues, they are conversion this type of the cycle. Then most of these complete of the C4 cycle, it is in the most than the C3 cycle it has in the plant, that is the chlorella plant. Now the C4 cycle is going to the mostly it has in the sugar cane. The plant name is sugar cane. Then most of it has in the plant, that is the almost of the plant it has the sugarcane plants, some maize, sugarcane, maize, and different type of the that is the corn. Sugarcane, maize, and corn plants they participate in the C4 cycle. So then why we come to the cycle, that is the C4 cycle? Then this is the mostly in research. The most famous scientist, that is the P.S. Ramaswamy Garu. Then it's going to the research in this type of the C4 cycles. Then the C4 cycle that has the mostly prevalent and researched kind of the cycle. It has most of the branch anatomy, depend on the branch anatomy, because in the C3 cycle they don't have any type of the it involving completely reaction mechanism, that is that or 12 types of the reaction mechanisms. But now then this kind of the cycle is involving the Hatch-Slack cycle, is involving in the branch anatomy. Because of the branch anatomy, that is the C3 it has in the corn, maize, or different types of the plants is involved in the branch anatomy, that mostly it kind of then the involvement of these of the grass plants is having only branch anatomy. Okay. Then now the question, why this cycle is in C4 cycle? Why we are mentioned in this cycle, C4 cycle? So that cycle it has most of among or overall of the cycle. Then the carbons are mentioned in 4 carbons. Every time it has in 4 carbons. That's why we call it the C4 cycle. That is the thing. Then now going to the pathway, it is the plants. It's completely it has then the tropical region of the plants in the that of the subtropical or the tropical region of the plants. Or that is the grass plants mainly they involve in the tropical or the subtropical type of the plants. So these are the region or these plants is tropical plants as well as subtropical plants. And subtropical plants. So the main of the plants, these are activity in some kind of that is the found of the plants most of the plants it has in the world, that is the sugar cane or any kind of the plants. Generally, it's like in the then another kind of the plants, that is the different of in the Amaranthaceae family, that is the Amaranthus is also involving in the C4 plants. Then it has the pathway is two kind of the pathway. Then the photosynthetic cells. It has involving in the completely in mesophyll bud cells or mesophyll sheath. It has complete involved in the mesophyll sheath. Then bundle sheath and mesophyll sheath. So this way the C4 cycle pathway is involving in the two ways, that is the mesophyll sheath as well as bundle sheath. Then mesophyll sheath and bundle sheath. So then this kind of two methods then involving in finally it is also involved in the C3 cycle. Then finally it is involved in C3 cycle also. Then it moreover often the it is involved the first it has in the CO2, that is the carbon dioxide in mesophyll is accepted. Then it is a that means it has in the bicarbonates is lost. Then the two carbons is lost. Then it involve of the primary carbons as well as then it most of it has in the primary carbons. Then accept of the cells, that is the first compound of the C4 cycle is, that is the pyruvic acid. Okay, the first compound is pyruvic acid. Then at the same time then it has also oxaloacetic acid. Okay, then the pyruvic acid it is converted to the oxaloacetic acid. So now the involvement of the two ways of the cell, it is now the first, now it has in the mesophyll cells. Then here is bundle sheath. Bundle sheath. Then the involvement of it has in the CO2 is given to the outside of the CO2. Then it has most of the CO2. Then it involve. Then or it is the CO2 is converted into the oxaloacetic acid. Then mesophyll cells. Okay. Then oxaloacetic acid. Mesophyll cells. Now going to bundle sheath. Then oxaloacetic acid, OAA. Then it gives to completely the mesophyll cells. It has in the CO2, oxaloacetic acid. Then oxaloacetic acid is converted into the malic acid. Convert into the malic acid. Then the enzyme or it is the another kind of the way, that is the involving of that is the phosphoenol pyruvic acids. phosphoenol pyruvic acids. So first of all, it is the oxaloacetic acid is gives to the oxaloacetic acid is gives to the malic acid. Okay. Then the enzyme is the malic dehydrogenase enzyme in the presence of malic dehydrogenase enzyme. Malic dehydrogenase enzyme. So here it complete then the malic acid when the malic acid is used to the it is also insert to the weight than the bundle sheet. Then it is 2 moles of the malic acid. Then it is going to the malic acid. Okay, bundle sheet also is given to 1 molecules of the malic acid. Okay, right. Then this malic acid to this malic acid, the use of the NADP plus H, then it is the use of in presence of the enzyme NADPH. NADP plus H plus. That means here also one kind of the inorganic phosphate IHOH. Then it's again involved in the malic acid. It gives to the pyruvic acid. Then the malic acid is gives to pyruvic acid. It is gives to pyruvic acids. To pyruvic acid. Then it has in the presence of the dehydroxylase enzyme. Dehydroxylase enzyme. Then NADP plus H plus is converted into complete in this reaction, that is the NADPH. Okay. Then the pyruvic acid, it is when the malic acid is split out of the CO2s. Then the CO2 is converted into the first product of the RUBP lipidose. RUBP. So this RUBP is present in the which kind of the cycle? That is the C3 cycle. So now it gives to the phosphoglyceric acid. Then RUBP is gives to the phosphoglyceric acid. Okay. Then the phosphoglyceric acid is converted again RUBP. Okay. Then the RUBP is converted to the phosphoglyceric acid. Again phosphoglyceric acid is converted to the RUBP. So this is the involved in the C3 cycle. Okay. Then it is involving in the C3 cycle. Then here of most of the bundle sheets, then the pyruvic acid is also is converted into the another molecule of pyruvic acids is into that is the mesophyll cells. Then this is also again in the mesophyll cells. Then pyruvic pyruvic acid. So in this type of the pyruvic acid enter into the mesophyll cells. This one is the pyruvic phosphate dikinase. Then it is a presence of the phosphoenol pyruvic acid PEP. It is used to the PEP. Phosphoenol pyruvic acid. Phosphoenol pyruvic acid. It is used to the phosphoenol pyruvic acid. Phosphoenol pyruvic acid. So then this kind of the phosphoenol pyruvic acids, then PEP is carboxylase. Then this one is the carboxylase. This is the carboxylase. So here kind of then the 12 moles of the NADP. NADP. Then it is involving into the 12 moles of NADPH. 12 moles of NADP plus 12H. Then here of the 30 ATPs and 30 ATP. These are in the inorganic phosphate, that is the I P 30 ADP plus 30 ADP plus 30 inorganic phosphates. Inorganic phosphates. So this kind of reaction it has in the pyruvic phosphate dehydrogenase. Then here the enzyme pyruvic phosphate dehydrogenase. Then the involvement of it has in the, that is the converted, then the it has mostly it has in the six reaction steps only. Then involved. It has the beta carotenes to dehydroxy acid cycles. Then involved also. Then it one of then the phosphoenol pyruvic acid is converted to tricarbonates. Then it is converted into the oxaloacetic acid. Then oxaloacetic acid is more over often the oxaloacetic acid in the presence of it has in the sheath, that is the involvement of the mesophyll cells to the bundle cells. Then here the product of the malic acid, then again that kind of the malic acid is produced in the bundle sheath also. Then pyruvic acid. Most these reactions it has completed in the type of the reactions is completed in almost all, that is the reduced to the malic acid. Then in presence of the enzyme, that is the malic dehydrogenase enzyme. Okay. Then again it is converted into the malic acid in bundle sheet that has the dehydrogenase enzyme. Then it has the involvement of the NADP plus H plus. Then NADP plus H plus. The moreover of it has utilized into the carbon dioxide. Then it convert into the RuBP. Because of carbon dioxide, that is the moreover of the C3 carbons, then the C3 carbons is present in RuBP, ribulose bisphosphates. Then the ribulose bisphosphates is again converted into the phosphoglycolic acid. Then again phosphoglyceric acid is converted into the regular bisphosphate. This reaction mechanism we can observe in the C3 cycle. Then the bundle sheet is connected into the C3 cycle, that is the Kelvin cycle. Okay. Then the reversible of the pyruvic acid is going on completely in the mesophyll sheath. Okay. Then the mesophyll sheath is also produced the pyruvic acid. Then the pyruvic acid, it is converted into the phosphoenol pyruvic acids. Then in the presence of the phosphophenol, the pyruvic phosphate dehydrogenase enzyme. In the presence of the pyruvic phosphodehydrogenase enzyme, it is converted into the phosphoenol pyruvic acid. Then it has moreover of the utilized of the 30 molecules of the ADPs plus 30 of inorganic phosphates. Then 30 of inorganic phosphates, then the ADP is utilized in the inorganic phosphates, then it converts into the 30 moles of the ATP. Okay. Then so here most of them the utilize for ATPs, they cannot remaining or they cannot storage of the ATPs is not here. Okay. Then moreover of the how much of then the ATPs are involved, ADPs are involved, that kind of the ATPs are produced. Then here most of it has the C4 cycles, the slack cycle in the presence of the enzyme, that kind of the enzyme moreover of the ATPs, that is the kind of the define of the 12 moles of the NADP in presence of the 12 moles of the NADP plus H plus is also required. Then how many NADPs are required? And sometimes they are asking in the practical examination viva part of view or any kind of the competitive exams is also. Then how many NADPs are involved in the C4 cycles? Then the blind answer, that is the 12 moles of the NADPs, that is enough. Then here the formation of the how many formations of the ATPs? Then going to the answer, 30 ATPs is formation in the C4 cycle. That is the only standard answers. It has the very we can easy answer, that is the 12 moles of the NADP. The required of NADPs is 12 moles. Then the formations of ATPs is 30. It is the simple kind of the answer. So then here after the molecule that has can be completed of moreover often that it has involved in the branch anatomy. Then the most of the that it has involvement of the branch anatomy, it has in the branch then the branch anatomy, then the most of the C4 cycles is provided the kind of the bundle sheet surrounding is it has in the branch anatomy. Then branch anatomy, that means the round of the sheath, they don't have any interspaces, that is called the branch anatomy. So for example here it is the branch anatomy, that is the sheaths is all involved in the 2 layers of the epidermis layer, then okay upper epidermis layer, okay. Then here moreover of the below that layer it is called then epidermis. Then the cells is completely surrounded of the another kind of the cells. Then that of Then it has some type of the stomatostructures. Then stomatos. These are the stomata. Then here the xylem. Then mesophyll cells. These are also mesophyll cells. Then this one is the lower epidermis. Lower epidermis. So then the branch anatomy is most of it has involving the bundle sheath surrounding of the sheaths, that is the known as then bundle sheath surrounding in the vascular bundles. Okay, vascular bundles means the presence of the xylem and phloem. Then the presence of the xylem and phloem, then vascular bundles is most, it is called to the branch anatomy. That means branch means it has in the width, then surrounds complete, it has in the round surrounding. Then the chloroplast, it has most data, that is the mesophyll cells. Then the mesophyll cells, most of it has in the chloroplast. Then these are the chloroplast. Then the most of the chloroplast in mesophyll cells. Moreover of the mesophyll cells than the chloroplast, it complete, it has which, where we have the presence in the kind of the this one, that is the PS1 and PS2, that is the photosystem 1 and photosystem 2. Because of them, most of the system it has utilized the chloroplast. Then the chloroplast is utilized in photosystem 1 and photosystem 2 reactions. So it most of occurrence in the, mostly it has in the reaction mechanism, that is the phosphoenol pyruvic carboxylations. Then the phosphoenol pyruvic carboxylations of the cells is longer cells of the mesophyll cells. Then the branch anatomy is completed, we can see and we can observe only the maize and corn kind of the plants complete. Then that kind of plants is used to the completely. Then this type of anatomical structure. Then it has then the reaction of structure or reaction mechanism. It's going on that of the carboxylase kind of the activity. Then this activity is going on completely PS1 and PS2 reaction centers. So these are the large size of the plants is chloroplast. Most of its that kind of the environment sheet. Then the chloroplast, moreover, then it has the leaf, then mesophills and then the bundle sheath. So its fixations of the chloroplast of bundle sheath help in the carboxylations of the formations. Then the size of the complete it has in the moreover in granas. Then mesophyll cells, bundles. So these are the generally in the C4 cycle. What is the significance of the C4 cycles? Then the significance is more over the environmental, it has the provide of the stomatos. The number of stomatos is open kind of the stomatos. Then it has closed type of the stomatos. The water is evaporation, that is called to the guttation type of the water. The guttation, then the leaf edges of the leaf, it is the guttation of the water. This one is the guttation. So here most of the water when the environment is involved, that is the prevent of reductions of the CO2 is the carboxylation. Then the so much of it has in the C4 plants, most of it has in the higher rate of the photosynthesis. Then these are the mostly high rate of the photosynthesis is a comparison between the C3 cycle. Then again carboxylations is also mesophyll and bundle shape. Then CO2 is released complete during of the respiration process. So these are the general kind of the C4 cycle, branch anatomy and the significance of the C4 cycle. Okay, thank you everyone.